as their RBCs become sickle shaped. As their RBCs become sickle shaped, that is half moon shaped only under abnormally low O2 concentration. That is half moon shaped only under abnormally low oxygen concentration. In this representation of pleiotropy, parents phenotype sickle cell carrier crossed with sickle cell carrier. Both the parents are sickle cell carrier. Then genotype are HbA, HbS, HbA, HbS. Then gametes HbA, HbS, HbA, HbS. Then offsprings are produced, that is HbA, 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 HbS, HbA, HbS, HbS, HbS. These are the four product, and that offspring shows the phenotypes HbA, HbA, normal, only one, HbA, HbS, HbA, HbS, sickle cell carrier, these are two in number. These are two in number. And sickle cell anemic dies, that is only one, HbS, HbS. Means that offspring shows the ratio 1:2:1. The homozygotes with recessive gene HbS, however, die of total anemia. Thus, the gene for sickle cell anemia is lethal in homozygous condition and produces sickle cell trait in heterozygous carrier. The different expressions are produced by single gene. That single gene show the different expressions. A marriage between two carriers will produce normal one, carriers 2, and sickle cell anemic children 1 in ratio 1:2:1. Sickle cell anemics die leaving carriers and normals in the ratio 1:2. The heterozygotes or carriers can be identified by microscopic examination of blood. Chromosomal theory of inheritance. Gregor Johann Mendel published his work on inheritance of traits in 1866, but for some reasons it remained unnoticed or unrecognized till 1900, as communication was not easy in those days. His work was not widely recognized. His approach of using mathematics and statistics to explain biological phenomena was totally new and unacceptable to then biologists. As continuous variations were observed in nature, Mendel's concept of factors or genes as stable and discrete unit which control the expression of characters, and that a pair of alleles did not blend with each other, was not accepted by his contemporaries. He also did not know the physical location of the factors or genes in the gametic cell. In 1900, three scientists, Hugo de Vries, Correns, and von Tischermack, independently rediscovered Mendel's work on the inheritance of traits. Walter Sutton, along with Theodore Bohri in 1903, studied the parallel behavior of Mendel's factor or genes and behavior of chromosomes at the time of meiosis. Based on these observations, chromosomal theory of inheritance was put forth by Sutton and Bohri. Based on these observations, chromosomal theory of inheritance was put forth by Sutton and Bohri. This theory identifies chromosomes as the carrier of genetic materials. This theory identifies chromosomes as the carriers of genetic material. This theory states that The chromosomes are present in pairs in somatic cells. This theory states that the chromosomes are present in pairs in somatic cells during gamete formation. Homologous chromosomes pair segregate and assort independently during meiosis. During gamete formation, homologous chromosomes pair segregate and assort independently during meiosis. Thus, each gamete contains only one chromosome from pair. Nucleus of gametes contains chromosomes which carry all hereditary traits. Male and female gametes, sperms and eggs carry all the hereditary traits. They are the link between parents and offspring. They are the link between parents and offspring. The fusion of haploid male gamete and haploid female gamete restore the diploid number of chromosome of the species. Here we have to understand there are link between parents and the offsprings. The fusion of haploid male gametes and haploid female gametes restores the diploid number of chromosomes of the species. Chromosomes. Chromosomes are filamentous bodies present in the eukaryotic nucleus. The term chromosomes was coined by W. Wilder in 1888. The size of chromosome varies from species to species. Each metaphose chromosome varies from 0.1 to 33 mm in length and 0.2 to 2 mm in thickness. Chromosomes are visible during cell division. They are capable of self-replication and play a vital role in heredity, mutation, variation, and evolutionary development. They are capable of self-replication. Chromosomes are capable of self-replication and play a vital role in heredity, mutation, variation, and evolutionary development in eukaryotic species. Chemically, eukaryotic chromosomes are made of DNA. Eukaryotic chromosomes are made of DNA, histone, and non-histone proteins. It means that chemically eukaryotic chromosomes are made of DNA, histone, and non-histone proteins. Function of chromosome. First, chromosomes are carriers of heredity. Number of chromosomes. The number of chromosomes is specific and constant for a particular species. Therefore, it is of great importance in the study of phylogeny and taxonomy of the species. The term ploidy speaks for the degree of repetition of the primary basic number of chromosomes, that is X, in a cell. When the chromosome number in a cell is the exact multiple of the primary basic number, then it is called euploidy. What is mean by euploidy? When the chromosome number in a cell is the exact multiple of the primary basic number, then this is called euploidy. Euploids include monoploid, haploid with one set of chromosome where X is equal to n, diploids 2n, 2 sets of chromosome, triploid 3n, 3 sets of chromosome, tetraploids 4n, 4 sets of chromosomes, and so on. When the chromosome number is not the exact multiple of the haploid set is aneuploidy. When the chromosome number is not the exact multiple of the haploid set is aneuploidy. Aneuploidy is either addition or deletion of one or more chromosome. Aneuploidy is either addition or deletion of one or more chromosome to the total number of chromosome in a cell. Here this variation in chromosome number, ploidy divided into euploidy, aneuploidy. Euploidy shows monoploidy one set of chromosome, diploidy, two set of chromosome, polyploidy, 3n, 4n, 5n, etc. etc. And in aneuploidy, hypoploidy, hyperploidy. In hypoploidy, monoswami, nulliswami. Monoswami 2n minus 1, nulliswami 2n minus 2. In hyperploidy, trisomy, trisomy 2n plus 1, tetrasomy 2n plus 2. That is the variation in chromosome number in ploidy. Structure of chromosomes. Chromosomes are best visible under microscope when the cell is at metaphase stage. When the cell is at metaphasic stage, the chromosomes are best visible under the microscope. We have studied in 11th standard. It is because at this stage chromosomes are highly condensed. Typical chromosome consists of two chromatids joined together at centromere or primary constrictions. Primary constrictions consist of a disc-shaped plate called kinetochore. Primary constriction consists of a disc-shaped plate called kinetochore. It is at the kinetochore spindle fibers get attached during cell division. Spindle fibers get attached during cell division. Besides primary constriction, some few chromosomes possess additional one or two constrictions called secondary constrictions. Besides primary constriction, some few chromosomes possess additional one or two constrictions called secondary constrictions. At secondary constriction, first nucleolus becomes organized during interphase. A satellite body, SAD body, is attached at secondary constriction second in very few chromosomes. Each chromatid in turn contains a long, unbranched, slender, highly coiled DNA thread called chromonema. Each chromatid in turn contains a long, unbranched, slender, highly coiled DNA thread is called chromonema, extending through the length of chromatid. Chromatid consists of double-stranded DNA molecule. Chromatid consists of double-stranded DNA molecule which extends from one end of chromosome to other. This is the structure of chromosome that shows the telomere. Then chromomeres inside, then secondary constriction, primary constriction, then chromonemata, then nucleolar organizer, then nucleolus and satellite. And A chromonemata are shown there, then in the middle centromere is shown, and there is matrix is present there. This is the parts of chromosome in A diagram, and that B diagram shows the secondary constrictions and other parts of chromosomes. That is the structure of chromosomes. Depending upon the position of centromere, it means that where the centromere is present, then that is the type of chromosome. Depending on the position of centromere, there are four types of shapes of chromosomes: acrocentric, J-shaped; telocentric, I-shaped; L-shaped, capital L-shaped, submetacentric; and metacentric, V-shaped. These are the four different types of chromosomes depending upon the position of centromere. The ends of chromosomes, that is chromatids, are known as telomere. Telomere is the end of chromosome there. This is the telomere. Sex chromosome. The chromosomes which are responsible for the determination of sex are known as sex chromosome. Allosomes. Human being and other mammals have X and Y chromosomes has 6 chromosomes. In human chromosomes, X and Y chromosomes are called 6 chromosomes. X chromosome is straight, rod-like and longer than Y chromosome. X chromosome is straight, rod-like and longer than Y chromosome. X chromosome is metacentric and Y is acrocentric. X chromosome has large amount of euchromatin and small amount of heterochromatin. Highly condensed region. X chromosome has large amount of euchromatin, short condensed region, and small amount of heterochromatin, highly condensed region. The chromatin has large amount of DNA material, hence genetically active. Euchromatin has large amount of DNA material, hence euchromatin is genetically active. Y chromosome has a small amount of euchromatin and large amount of heterochromatin, hence it is genetically less active or inert. Both X and Y chromosomes show homologous and non-homologous region. Both X and Y chromosomes show homologous and non-homologous regions. Homologous regions show similar genes. Homologous regions show similar genes. Non-homologous regions show dissimilar genes. Homologous regions show similar genes. Non-homologous regions show dissimilar genes. Crossing over occurs only between homologous regions of X and Y chromosome. Crossing over occurs only between homologous regions of X and Y chromosomes. Non-homologous regions of X chromosome is longer and contains more genes that of non-homologous region of Y chromosome. X-linked genes are present on non-homologous region of X chromosome. X-linked genes are present on non-homologous region of X chromosome. Y-linked genes are present on non-homologous region of Y chromosome. This is the structure of X and Y chromosomes in human. At the basal side, there are two regions of that X and Y homologous regions, that is showed by blue colored, and in the middle side centromere is present in both X and Y chromosome. X chromosome is longer, Y chromosome is shorter, and at the apex short length of non-homologous part in Y chromosome and extended longer parts of non-homologous region in X chromosome. It means that non-homologous region of X and Y chromosome, and in the below that centromere is present there, and at the base the similar